Cell membrane potential, nervous system. A cell membrane is usually electrically charged. You have both positive and negative ions. And when you have that electrical charge on the cell membrane of a nerve cell, you say it's polarized, so that the inside of the membrane is negatively charged with respect to the outside of the membrane being positively charged. The membrane potential then is resulting in this charge due to the unequal distribution of ions on both sides of the membrane. So that resting neuron cell membrane, or the cell membrane resting potential, is polarized or electrically charged because you have those differences. So what we see is the potential difference, also known as the PD, exists across this resting membrane, and the potential difference ultimately is the difference in electrical charge between 2 points, that is 2 points on the membrane from the extracellular or outside the cell compared to the intracellular or inside the cell. The ions that we're looking at here are sodium and potassium ions. Both are cations because they contain a positive charge. So those positive potassium ions, those are intracellular ions, and the sodium ions are extracellular ions. And basically these two ions are going to travel across the nerve's membrane using ion channel proteins through a process known as active transport. Now, this specific type of active transport, as these ions are traveling from areas where they're in lower concentration and going to be pumped into areas of higher concentration, is an example of the sodium-potassium pump. So the resting membrane potential, the RMP, of a neuron results from this distribution of ions across the cell membrane. So we have high potassium ions on the inside, high sodium ions on the outside. You also have some negative ions or anions in there. You have chlorine as an anion. That will be highly concentrated on the outside. And then you also have negatively charged proteins. If you look at protein structure, you have negative regions of proteins or ions, anions that are also highly concentrated on the inside of the nerve cell. So these ions and their concentrations are maintained by the active transport mechanism, such as the sodium-potassium pump, the ATPase pump, etc. So that is all types of active transport. So looking at the resting potential, or the RMP, resting membrane potential, of a nerve cell, basically the resting membrane potential of a nerve cell is measured in millivolts. So you have a 70 millivolt difference from inside to the outside of the nerve cell. The resting membrane potential of a nerve cell is measured to be about negative 70 millivolts. And basically, when you have this charge on the membrane, it is said to be polarized. So as long as the resting membrane potential in a nerve cell is left undisturbed, it remains polarized. However, in order for that impulse to be started or propagated in a nerve cell, the resting membrane potential needs to be disturbed. So inside that cell, you have a negative relative charge compared to the outside of the cell. And this distribution of ions inside versus outside the cell is what causes this resting membrane potential. And that sodium and potassium pump is going to restore all this. So if we look at the diagram, you probably can't see it very well here. In this first picture here, what you have are low potassium ions, and those low potassium ions are found outside the cell, and you have high potassium ions inside the cell. Here you have high sodium outside the cell, and your low sodium is inside the cell. So what's going to happen, if you imagine the cell before the membrane potential is established, the concentration gradients are such that the fusion of potassium ions outside the cell is going to exceed the diffusion of sodium ions into the cell, and it's going to cause a net loss of positive charge from the cell. Now down here you have, in the second picture, you have a negative 70 millivolts membrane potential there. So the net loss of those positive charges from inside the cell has left the inside of the cell membrane slightly negative compared to the outside of the membrane, which is left slightly positive. This difference is the electrical potential difference, that PD. So this electrical potential difference is measured as negative 70 millivolts in a typical neuron, and this is called the resting membrane potential. Now down here in the third cell, which you see here in the diagram, what you see is the sodium-potassium pump at work here. So you can see sodium ions are going to leave the cell where they're in low concentration, and potassium ions are now going to come into the cell where they're highly concentrated, and then you still have that negative 70 millivolts in the axon region. So with the membrane potential established, the sodium diffusion into the cell is now aided, and potassium diffusion opposed by the negative charge on the cell as the potassium ions leave. But the action of the sodium-potassium pump balances the movements, and the result is a concentration of these ions and the resting membrane potential are maintained. So when you look at local potential changes, it is caused by those various stimuli. You could have temperature changes, light, pressure changes. So environmental changes do affect the membrane and the potential by opening a gated ion channel. And these channels can be, one, chemically gated, they can be voltage gated, or third, they can be mechanically gated. So when you look at those local potential changes, the resting membrane potential of negative 70 millivolts can be distributed disrupted or changed in one of two directions. It can become more negative, which we would say is hyperpolarization, or it becomes less negative as it goes towards zero or towards the positive end, which will be depolarization. And you can see that all right here when you look at the local potential changes. So environmental changes can cause gated ion channels to open. As the ions then flow through the membrane potential is going to change. If the membrane potential becomes more negative, it is hyperpolarized, and we'll see this later in a diagram. If a membrane potential becomes less negative, it is going to be depolarized. And it is graded or proportional to the intensity of the stimulation reaching that threshold potential. And we'll look at that threshold potential a little bit later. So when you reach that threshold potential, it triggers voltage in the gated channels and causes them to open, which then causes an action potential. Typically, when you look at the threshold potential, it's probably around negative 55 millivolts. So you have that negative 55 millivolts on the membrane then. So you go from that negative 70 to a negative 55 millivolts, and then you reach that threshold stimulus is a positive 15 millivolts on the membrane. So that subthreshold depolarization will not result in that action potential. And you can see that associated here where you have neurotransmitter. Here's the presynaptic neuron coming in, and there's the chemically gated ion channels. So you can see as you go from negative 62 millivolts to negative 55 millivolts, and negative 55 millivolts, you have reached that threshold potential. And once you reach that threshold potential, the resting membrane potential of a neuron becomes depolarized, and it starts to open up those gated channels. So here you can see sodium ions now coming in the second, in picture B, you can see sodium ions now entering in along the axon as that voltage starts to go down the axon of the neuron there. where those, really quick, where those sodium ions are going to come in, it's going to follow it down. This is called the trigger zone. And their opening is what's going to trigger the action potential. So here is what action potential is. At rest, the membrane is polarized, so the resting membrane potential is negative 70 millivolts. As you have ions moving in, all right, so the potassium ions are there, that threshold stimulus is reached. So basically the cell membrane is going to be depolarized, so it's going to become less negative. It's starting to move towards the positive end or closer to zero. And that is when you have that threshold stimulus. So the cell membrane of the neuron must be depolarized to approximately negative 55 millivolts in order for certain protein channels to open and therefore start an action potential or an impulse. So once that threshold stimulus is reached at about negative 55, those sodium ion channels are going to open up and it's going to further depolarize the membrane towards zero. And here you can see that threshold stimulus going down, the sodium channels open up, and sodium ions start to come into the region there. So this is the region where this is occurring, is called the region of depolarization. So as we go further, the resting membrane of a neuron is depolarized, negative 55 millivolts. That causes that threshold potential to be released. The threshold potential of the neuron is now at negative 55 millivolts. And therefore you have that threshold stimulus, which is about a positive 15 millivolts. When that threshold potential is released, you have the rapid opening of sodium ion channels that are going to result in rapid depolarization. And it's going to cause a reversal of the membrane potential to go to plus 30 millivolts. So potassium ions leave the cytoplasm and the membrane is going to now repolarize itself. And in repolarization, basically what's happening, this event is called the action potential. And the action potential represents the start of an impulse on that neuron. And then what will happen is the potassium channels open while the sodium channel's closed, and then you start to repolarize the nerve cell, and that's when you're going to have that recovery back to the resting membrane potential of negative 70 millivolts. This all happens very quickly. It happens in about 1/1000th of a second. So you have the resting membrane potential here, and if you look at the diagram here, you can see negative 70 millivolts. The sodium channel opens, potassium channel is closed, and now you start to go reach that threshold, and then it starts to repolarize itself, and then that's where you have the potassium channels opening and the sodium channels closed, and then you come down below that negative 70 millivolts, and because you are further negative, that is hyperpolarization, until eventually you restore it back to its resting membrane potential. So it'll eventually come back to that negative 70 millivolts there in the diagram. So the trigger zone itself, action potentials at the trigger zone. The trigger zone is the first part of the axon that contains many voltage gated ion channels of sodium, and those voltage gated sodium channels are going to open in response to the threshold. As the sodium moves in, the membrane depolarizes until it reaches positive 30 millivolts. That's the action potential. The sodium channels closed, potassium channels open, potassium ions move out, and the membrane repolarizes.